So this is the last part, part 4, where we go into a little more detail on the assay and some of the recent findings. So I've just described the 4-color probe configuration, and of course not all deletions of PTEN are the same. That's one thing I should stress. There are different variations on the theme. In addition to having one copy lost, that's hemizygous deletion, or both copies lost, that's homozygous deletion. So this is from a paper of ours that's we hope will be appearing later this year. I should just to keep you alert, in this configuration PTEN is green, it's not red as you just saw in the previous presentation. For reasons I won't bore you with, we switched colors from red to green. So if you look at this area here, all the nuclei here in general have 2 copies of PTEN plus the flanking probes. It's quite easy to see the amount of colors coming from this region. But in this region here, you're seeing loss of both of the green signals. So this is a homozygous deletion in this region. And there are various combinations of things that we've seen that are reported in this paper. So this is a hemizygous deletion. So you can see just one spot there. Another hemizygous deletion, it's lost the red and the green signal. The other chromosome seems to be okay. Another hemizygous deletion here, and so on. So a number of different things can be seen, and these are shown, or this is just to show you the difference between the region with a homozygous deletion and the normal cells here. I should point out that this was an area of cancer, and the pathologist had made the delineation of the normal cells in the prostate gland here. So this is the reason that you're seeing a deletion here and not here. So a number of different things have been seen. So if the nuclei don't have a deletion, this is a typical sort of pattern you see. Then if one copy is lost by a small deletion, so it's keeping the 2 flanking probes A and B but losing PTEN, this is what you'd see. Similarly, another hemizygous deletion, but this time the red probe B and PTEN have gone together, so you're seeing less signals but losing green and red. And this is a 1 copy loss, but it's a very big deletion. It includes both probe B and probe A. So on one chromosome everything is there, on the other we've only got the centromere. This is the most common homozygous deletion that we see. It's a smallish deletion just involving PTEN and keeping probe A and B. So this is something that we've seen as the most frequent homozygous deletion. Another homozygous deletion, this time one of the chromosomes has similar to this one. The other one has lost all probes except the centromere, so a big second deletion there. So those are some of the things that one sees when one's doing scoring. In this particular paper, 330 prostate cancers were examined. It was on a tissue microarray, and about 40% of them, 132, had a deletion. And this is the breakdown. So the hemizygous deletions, the most common one was that one that I just mentioned, keeping probe A and B, losing PTEN. Second most common, loses PTEN and probe B, and so on. Similar sort of distribution in the homozygous deletions. The 2 copy loss is shown in red. So the most common is this one that's just PTEN, but then the second most common extends into probe B. And then you see other combinations, so that a small deletion and a big deletion, a small deletion and a big deletion going that way, and then here some very rare events that we've seen. So one can map it out. The take-home message is that you don't always see the same type of deletion. Now, there's a lot of interest, I think, in scanning, and I'm working with applied spectral imaging, and there are other companies I know that are also doing this kind of scanning approach, and so one can collect nuclei within these sort of thumbnails and do some automated spot counting. And I think that's an area that we're going to see a lot of changes over the next 12 months. So this is a very exciting area to watch. At the moment, I think as one is developing the assay, it's best to have an experienced analyst score by eye and score on sheets. I think it's still not quite there for scanning, but it's not far off. And this is an example of a homozygous deletion detected using the scanning. So you can see PTEN is green, so you're seeing the flanking probes but losing the 2 green, and the counter goes through and spot counts. So this is from one of our recent papers where we just try to put it all together as a sort of schematic. So we think that the single copy loss, the hemizygous deletion, is a fairly early event, and as I said in the first presentation in this series, that we've observed that in PIN cells. We know that the function of PTEN protein can lead to proliferation and also instability. So changes such as the TMP-ETS fusion event could also occur around this time. And then I think when prostate cancer occurs and progresses, one loses a second copy of PTEN, and this seems to lead to a more aggressive phenotype. And so I wanted to just finalize this session with a little discussion on that. This is a study that was published in 2009 and follow-up 2010, looking at castrate-resistant prostate cancer. And so we did an analysis again using tissue microarrays and did a combination of fusion gene analysis by FISH, PTEN by FISH, androgen receptor by FISH, and then some immunohistochemistry. And this is from the paper. I won't spend too much time on it. The paper is just here. But we saw a strong association with loss of PTEN, phospho-AKT, phospho-mTOR, and other biomarkers in this PI3 kinase axis. In addition, a strong association with androgen receptor amplification. Within this cohort, we saw some tumors with a mixture of homozygous and hemizygous deletions. And so when we see a mixture like that, we classify the tumor as having lost both copies of PTEN, because this is obviously clinically the most significant and the worst finding. So I think in terms of how one treats patients with this kind of thing, one has to consider these as having lost both copies of the PTEN gene. And this is just from the paper again, and you can see that within this cohort of the 23 men who died of prostate cancer specifically here, 3 out of 20 had no deletions, so 17 out of 20 had deletions. So this was significant within this cohort. You also see a significant association with the amplification as well. So, to summarize, the incidence of PTEN deletion in high-grade PIN associated with prostate cancer is something like 20%. There's very little data currently available on the clinical implications of these losses, but certainly I think this is accumulating quite quickly. The overall incidence of PTEN losses in radical prostatectomy samples ranges from 20 to 40%. And the loss of the gene is associated with earlier PSA recurrence, as I described, and also shorter overall survival. Loss of both copies of the PTEN gene occurs in about 5% of radical prostatectomy samples, but may be observed up to 40% in castrate-resistant prostate cancer and as high as 80% in metastatic tumors. So there's really accumulating evidence suggesting that loss of both copies of PTEN is a poor prognosticator. And there is this association between other types of genomic change, such as the fusion gene, MYC gain, and androgen receptor amplification. So that it's clearly one of the things that one can do by FISH, but there are other emerging genomic biomarkers that may be relevant. So I should finalize now by thanking my colleagues and my excellent laboratory team here in Kingston at Queen's University. And thank you for looking at these presentations. Thank you very much. Bye-bye.